Hello, we're going to go ahead and continue in the 3.2 notes. We're going to look at topic 2 in these notes. So in the last topic, we looked at oxidation reduction reactions and big picture what's going on during cell respiration. In topic 2, we're going to now go into the fun details of how this is all taking place. And so specifically in topic 2, we're going to look at the first few phases of cell respiration, which is glycolysis, pyruvate oxidation, and the Krebs cycle. So what we're about to look at is a very huge series of chemical reactions taking place that are going to ultimately help lead to this process of glucose breaking down in the presence of oxygen into CO2 and water being formed. And so when we're talking about cell respiration, usually by default we're referring to aerobic cell respiration, which aerobic means something that's using or requiring oxygen. It's an oxygen using process. And so when we say cell respiration, by default, I'm usually referring to aerobic cell respiration, which requires oxygen. There are anaerobic processes that can take place without oxygen, but that's going to be different, and we're not going to talk about that until later in the 3.2 notes. But for now, we're going to look at, okay, if there is oxygen available, how does this look like when there is oxygen, this process of cell respiration? And when there is oxygen, oxygen will be the person, his role is to be the final acceptor of those electrons that we are taking from glucose, just like we talked about in the last video. So that's the whole reason you guys are breathing right now, is just for oxygen to be the person accepting those electrons at the end of cell respiration to allow your body to continue making more and more ATP to power your body. So cell respiration, or aerobic cell respiration, it occurs in, well, we could break it into 4 stages. And so in these notes, we're going to look at these first 3 stages. This picture right here gives you a nice overview of what we're about to talk about. So starting with glycolysis, we'll look at what that looks like. That takes place in the cytosol. So this is outside of the mitochondria, this process called glycolysis. And in glycolysis, that's a process that basically almost all cells on this planet do. Basically every type of living thing on this planet can do glycolysis because it doesn't even require a mitochondria. And like we discussed before in this class, not all cells have a mitochondria. Prokaryotic cells don't have a mitochondria, but prokaryotic cells can even do glycolysis because you don't need a mitochondria to do it. It's basically, it's one of the unifying characteristics of all life on this planet is that we all can do glycolysis. And that's how this whole thing gets kickstarted, is with glycolysis. Now, if you do have a mitochondria, which this would apply to eukaryotic cells, and if oxygen is available, then we will continue on to these other steps, these other stages, starting with pyruvate oxidation and the citric acid cycle, which we'll talk about in this video as well, before leading to the grand finale, which is oxidative phosphorylation, which we won't talk about until topic 3, just to leave you guys in suspense how the story ends. Be a cliffhanger, but we're going to look at these first 3 steps. And so again, glycolysis is outside the mitochondria, and then pyruvate oxidation, the citric acid cycle, these take place inside the mitochondria. Specifically, those 2 processes take place in the inside of the mitochondria, the matrix of the mitochondria. So just to give you guys a quick refresher about the mitochondria, we talked about this in a previous unit, unit 2. It has a double membrane. There's the outer membrane, and then there's the inner membrane, which is all folded up. And then there's the intermembrane space, which is the space between the outer membrane and the inner membrane, like where my mouse is pointing. And then there's the matrix of the mitochondria, which is like the inside of the mitochondria, inside the inner membrane. This light yellow area, that's the matrix. And that's where pyruvate oxidation and citric acid cycle take place, which involves a bunch of chemical reactions and enzymes that are found in the matrix of the mitochondria. But we're going to start outside the mitochondria with glycolysis. So let's take a look at that. Sorry, my slides are frozen. Okay, I don't know what happened. Here we go. So in glycolysis, this is a picture that shows you what's happening in glycolysis. It involves 10 different reactions. There's 10 reactions taking place in glycolysis. Each of those reactions involves its own enzyme. We're not going to go into every single reaction. We're just going to go over and kind of a detailed overview, though, of what is happening in these 10 reactions. Until you guys take like a biochemistry course in college, you won't have to memorize a lot of the actual reactions. It gets pretty miserable pretty quick learning every single reaction and every single enzyme and every single chemical compound made in between each reaction. But we're just going to look at overall big picture what's going on here. And so glycolysis, let's just start off with the fact that we're going to ultimately start with glucose, which is a 6-carbon molecule. That's what we're showing right here with these spheres. It's 1, 2, 3, 4, 5, 6. We're just looking at the carbons. I want you guys to keep track of the carbons. There's 6 carbons in glucose. It's a 6-carbon molecule. I'm obviously not showing you the rest. This is just a simple depiction of glucose. It's C6H12O6. I'm not even showing the hydrogens or the oxygens. And also glucose isn't linear. It's like a ring structure, but we're just going to look at keep track of the carbons. We're starting with 6 carbons in glucose. And ultimately what's going to happen is during glycolysis, we're basically going to split glucose into 2 smaller molecules called pyruvate. And so pyruvate is a 3-carbon molecule as represented by these dots here. And we're going to end up with 2 of them at the end of glycolysis. So glycolysis literally translates to sugar splitting, glycolysis, sugar splitting. And that's exactly what we're doing. We're going to split some sugar. We're going to take glucose and split it in half. And you would think that would be super easy, but it actually involves 10 different reactions to do that. But that's what's going to happen. Don't get too overwhelmed by this picture yet. And like I said, this occurs in the cytoplasm outside the mitochondria, and it can be divided into 2 phases. There's the energy investment phase and the energy harvesting phase, or in this case, they call it the energy payoff phase, either way. Let's start with the energy investment phase. And the energy investment phase, basically what I need you guys to know is we're taking glucose and we're actually going to use 2 molecules of ATP to ultimately turn it into these 2 guys here, which are called G3P. And so we're going to take, if you guys look at what's happening in this picture, this glucose is going to turn into a molecule that also has 6 carbons. This is no longer glucose though, and it has 2 phosphates attached to it, a phosphate group over here and a phosphate group over here. Those phosphates came from ATP. So ATP has 3 phosphates, and the third one, like we talked about, he's like just trying to get rid of it. That third one pops off really easily so that ATP becomes ADP. And basically what's happening here is ATP is transferring that phosphate to glucose. So he's dropping off a phosphate to glucose, and then he's dropping off another phosphate to glucose. And now we end up with this guy, who is basically glucose, but with 2 phosphates. He has his own name. What's his name? We don't care. You don't need to know that for this class. But he's a very unstable molecule, very high energy. We just took a molecule that the universe doesn't like, and we created a molecule that the universe really doesn't like. This is a very unstable, high free energy molecule. And this molecule very quickly splits and easily splits through a couple of reactions into G3P, which is this guy right here, which has 3 carbons and a phosphate group. And so that's what's happening in the energy investment phase. It's called the energy investment phase because if you're looking closely here, we haven't even made ATP yet. We've actually done the opposite. We've used ATP. So far, we're off in the negative direction. Like we haven't even made ATP. We're actually using ATP to start off with. So we're net negative 2 ATP so far. Like we haven't even made ATP. We lost 2 ATP. So, but we got to do this first investment of energy to kickstart this whole thing. And so that's what's happening here. We're using, so just know, all you guys need to know is that in the energy investment phase, we're taking glucose and it's going to require 2 ATP molecules to turn it into these 2 molecules of G3P. You guys do need to know the G3P. So just know that you're ending with G3P here. He's a 3-carbon molecule. That's all I need you guys to know. So now we have these 2 molecules of G3P that we're ending with after the energy investment phase. And then once we have G3P, we can now start the energy harvest phase or the energy payoff phase. In the energy investment phase, the energy harvesting phase, we're going to take those 2 molecules of G3P and we're going to turn them into 2 molecules of pyruvate. That's ultimately what's going to happen. Now in the process of that happening, there's like 5 or 6 reactions taking place, which we're not going to go into detail, but overall what's going to happen is the G3P, there's going to be, if you just follow this picture, what's happening is a phosphate is being added to it. So we're going to add another phosphate. This one's just randomly floating around in the cell. We're going to find a phosphate and we're going to pluck it onto this guy. But now we have this guy. Who is this guy? We don't care. We don't need to know. But now he has these 2 phosphates on it. And then what's going to happen in a series of, this is actually 4 reactions that are going to take place here, but you don't need to worry about it. We're going to take this guy and we're going to take off those phosphates and we're going to give them to ADP. So ADP is going to swoop in and take one of those phosphates. We're going to transfer one of these phosphates to ADP. so that he becomes ATP. So we're going to give this guy who has 2 phosphates his 3rd phosphate back, and now he's ATP. And we're going to do that twice for this guy. So we're going to do 1 ADP becoming ATP and then another ADP becoming ATP. So then we're going to end up with 2 ATP molecules here. The same thing's going to happen over here with this G3P. We're going to have 2 ADPs swoop in and take those phosphates, and they're going to become 2 molecules of ATP. And then also in this process, we're going to steal our very first pair of high energy electrons. So here's NAD+. I told you guys about him. He's an electron carrier molecule, and in one of these reactions, he's going to swoop in and take a pair of high energy electrons that used to belong to glucose, this guy, and he's going to take those electrons and go away as NADH. So here's NADH. He's now holding on to that pair of high energy electrons. Same thing happened over here. And so we just stole our first 2 pairs of electrons. This pair of electrons was stolen, and then this pair of electrons was stolen. And again, whenever you see NADH, just know he's holding on to a pair of high energy electrons, and those electrons originated in glucose. Like that's where they came from before he started breaking down into these guys. And so overall, in the energy harvesting phase, when we take these 2 molecules of G3P and we produce these 2 molecules of pyruvate, we end up with making 4 molecules of ATP, 2 plus 2, that's 4 molecules of ATP that we made, and 2 NADHs. So we have an NADH over here who swooped in and has holding on to a pair of high energy electrons, and then over here. And so that's what's happening in the energy harvesting phase. And then you have pyruvate, which is a 3-carbon molecule that you need to know. So when I say pyruvate, you need to know, okay, that's what we end glycolysis with, and it contains 3 carbons. And so that's the gist here, the detailed gist of what's going on. So overall, during glycolysis, like the net production during glycolysis from beginning to end, from glucose to pyruvate, we net a production of 2 ATP because we were -2 before, and then we made 4. So you made 4, but you already used 2. So really, you just made a net of 2 ATP. So overall, we produced 2 ATP because we made 4, but we lost 2. And then we also end up with 2 NADHs. And then now we have pyruvate, no longer glucose, but this was glucose. So this is glucose now, but now it's pyruvate, okay? That's it. So you guys, there's some memorizing you got to do here, but I believe in you guys. So what we just did is we took glucose, turned it into 2 molecules of pyruvate. We had a net production of 2 ATP and 2 NADHs. That's all you need to know for glycolysis so far. And there's an energy investment phase and an energy harvesting phase. Okay, that's big picture what's going on. So, and in class, we're going to diagram a lot of this. You guys drawing out a lot of it is going to help you ingrain it in your head correctly. But try your best right now to start getting the feel for what's going on here and start memorizing some of this stuff, the big picture overall things going on so far. So that's glycolysis. All of that took place inside the cytoplasm of the cell. And now we're going to move on with the pyruvate. The pyruvate is now going to go into the mitochondria. So if there's a mitochondria, because you're a eukaryotic cell, and if oxygen is available, the pyruvate will be transported into the matrix of the mitochondria. So that's where we're headed next, into the mitochondria. Before we do that, though, real quick, I forgot. You guys need to know that the ATP that we just made here, these 4 ATPs that we made, those during glycolysis, those ATP were made through a process called substrate level phosphorylation. There's some different techniques to making ATP in the cell. Substrate level phosphorylation is one technique to make ATP, and that's how we just made the ATP just now. It involved an enzyme. There was an enzyme who took a phosphate from a molecule and gave it to ADP. And so there is a substrate, and we're using it to phosphorylate ADP to make ATP. And so that's this enzyme here who's helping that substrate to transfer that phosphate to ADP. So that's called substrate level phosphorylation. You can just memorize that that's how this ATP was made. Hopefully that kind of makes sense. I don't know. There's an enzyme basically taking the phosphate from this guy and giving it to ADP. And then another enzyme taking the phosphate from this guy and giving it to the other ATP. It's called substrate level phosphorylation. That's how we made that ATP. But anyway, now we move on to pyruvate oxidation. So now the pyruvate is going to go into the inside of the mitochondria. So now we're in the matrix of the mitochondria. And now what we're going to do in stage 2 here, it's called pyruvate oxidation. This is a really short process. It's actually just one chemical reaction that accomplishes a few different things. But basically what's going to happen is we're going to take the pyruvate, that we ended glycolysis with, and we're going to turn it into a molecule called acetyl-CoA. So pyruvate is going to become this molecule called acetyl-CoA. Now, acetyl-CoA is only a 2-carbon molecule. As you guys can see here, it has 2 carbons, no longer 3 carbons. So we just lost a carbon. So we went from 3 carbons in pyruvate to 2 carbons in acetyl-CoA. One of those carbons was lost, and that carbon that was lost actually was released or produced as CO2. So there was a molecule of CO2 made here, which has 1 carbon. And that was basically chipped away from this molecule, pyruvate. We chipped off a CO2 molecule who's carrying a carbon and 2 oxygens away. Again, we're not tracking the oxygens, we're just tracking the carbons right now. And so that CO2 molecule is, during pyruvate oxidation, gets produced, leaving us with only 2 carbons from those pyruvate, the 3 carbons that were in pyruvate. And also during this process, we stole another pair of electrons to make NADH. So here's NAD+, and he's swiping in like Swiper the Fox and taking some high energy electrons, becoming NADH. So now he's holding on to those high energy electrons that he stole from pyruvate, who used to be glucose. Really, like all of this started off as glucose. And then we added on this coenzyme called coenzyme A. It's just a small little molecule that gets latched onto to the pyruvate to become acetyl-CoA. Doesn't matter too much. So really just know in pyruvate oxidation, that sounded like a lot, but really you're taking pyruvate, you're turning it into acetyl-CoA, which only has 2 carbons now. A CO2 molecule was produced, which explains why there's only 2 carbons left. And we took a pair of high energy electrons to make NADH. Now remember, this is happening twice because we had 2 pyruvates. This is just showing you with 1 pyruvate, but remember there was 2. We ended glycolysis with 2 pyruvates. So we're going to keep track as if starting with 1 glucose, ultimately what's happening. So if you start with 1 glucose, you have 2 pyruvates, and each of those pyruvates is going to go through pyruvate oxidation and become 2 molecules of acetyl-CoA. So right here on the notes, I keep track for you guys. During the oxidation of both pyruvates, you're going to have 2 carbon dioxides produced, 1 from each pyruvate, 2 NADHs that stole a pair of high energy electrons, 1 from each pyruvate, and you're going to end up with 2 acetyl-CoAs, 1 from each pyruvate, right? So remember this is double, okay? So that's what's happening during pyruvate oxidation. Now you have acetyl-CoA, which is a molecule you guys do need to know. So acetyl-CoA, we went from pyruvate to acetyl-CoA. This can now move on to stage 3, the final stage that we're going to talk about in these notes. In stage 3, we're going to now enter the Krebs cycle, which is also called the citric acid cycle. This also takes place in the matrix of the mitochondria. But basically, pyruvate oxidation is just a way, sometimes we call it the link reaction. We have to take the pyruvate and turn it into acetyl-CoA before we can now enter the next step. So it's kind of like a transitionary reaction that helps link glycolysis with the citric acid cycle, the Krebs cycle. But anyway, now we have the acetyl-CoA. So here's the last stage here in these notes for topic 2. We're going to look at the citric acid cycle. The citric acid cycle is a series of 8 different reactions, almost all of which you don't need to know individually, but this picture kind of gives you a lot of details about, okay, well, here's what's happening. It's a cycle. And so here's acetyl-CoA. I'll just give you guys the gist of what's going on here. Don't get too overwhelmed. Here's acetyl-CoA, right? 2 carbons. We just saw him. We ended pyruvate oxidation with him. Oh, by the way, pyruvate oxidation, we call it oxidation, pyruvate oxidation, because pyruvate is being oxidized. Oxidized means losing electrons, and he was losing electrons. NAD plus stole some electrons from pyruvate. So NAD plus was being reduced into NADH, gaining those electrons, and pyruvate was being oxidized by losing those electrons. That's exactly what was happening there, if that helps you remember. Anyway, now we have acetyl-CoA, and what's going to happen, this 2-carbon molecule, and again, this is going to happen twice. So I'm going to show you, because again, we have 2 acetyl-CoAs now, and each acetyl-CoA is going to go into the citric acid cycle, and then the next acetyl-CoA is going to go into the citric acid cycle. So we're going to have 2 rounds of the citric acid cycle, what I'm about to explain, 1 for each acetyl-CoA that we just ended with. So if you look here, what's going to happen is there's this molecule called oxaloacetate. Oxaloacetate is a 4-carbon molecule, and it's going to join together in this to start this cycle. We're going to take acetyl-CoA and we're going to join it to oxaloacetate. So oxaloacetate and acetyl-CoA are going to join together. There's a reaction that joins them together. And when you join them together, you're going to end up with a 6-carbon molecule, 2 plus 4. You're going to end up with a 6-carbon molecule called citrate. This is citrate. Citrate is also called citric acid, which is the name of the cycle. So this is citrate or citric acid. It's a 6-carbon molecule, and this is us starting the cycle here. Okay, oxaloacetate, acetyl-CoA, they join together, and now we're going here. Here we have citrate, and now the cycle begins with citrate. And like I said, this is also called the Krebs cycle. The College Board usually refers to it by the Krebs cycle, so I'm going to start saying Krebs cycle instead of citric acid cycle. So in the Krebs cycle, you have citrate, and basically there's a lot of reactions that are going to take place. But all I need you guys to know is that overall, you just need to overall know what's happening. So like just memorize what's the things that are going to be made here. There's a lot that is going to end up happening here. But if you guys look, ultimately the citrate is going to become boom, 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 boom. It's going to go back to being oxaloacetate. Just know that, right? Which is why this is called a cycle, because you're going to start and end the cycle with the same thing. So citrate is going to go back to being oxaloacetate through these reactions, which is going to require then that citrate loses 2 carbons. If we have a 6-carbon molecule and we want to go back to oxaloacetate, which is a 4-carbon molecule, he's going to have to get rid of 2 of these carbons. And what's going to happen in the citric acid cycle is those 2 carbons are going to be chopped off and released as CO2 molecules. So we're going to release 2 CO2 molecules. You guys can see it right here and right here. 1 of them, 2 of them. So there's one of those carbons is going to be released as CO2, and then another one's going to be released as CO2. So you're going to lose those 2 carbons right there. So 2 carbon dioxides are made here, which explains why we are starting with 6 and then go back to 4 carbons. And then there's a bunch of electrons we're stealing. This is where we're going to really cash in and steal a bunch of electrons. So here's NAD+, he's going to swoop in and take a pair of electrons. Here's another one, he swoops in and takes a pair of electrons. Here's another one, he swoops in and takes a pair of electrons. So then we have 1, 2, 3 NADHs being made, taking those electrons, taking electrons, taking electrons. And again, ultimately, these electrons started from glucose. That's where all this came from, because that became pyruvate, and pyruvate became acetyl-CoA, and acetyl-CoA entered this cycle. And so those electrons are being taken here, here, and here by NADH. So we end up with 3 NADHs that are being made in the cycle. And then there's FAD. Remember him? I told you he's also an electron carrier molecule. This is basically his time to shine. This is the only time we're going to see him. But he swoops in and takes a pair of high energy electrons as well. Becomes FADH2. And that's going to happen once here in the cycle. And so we end up with 3 NADHs being made and 1 FADH2 being made. Each of those is holding on to a pair of high energy electrons that they stole. And then they're going to go take those somewhere else. We're not going to talk about that yet. And then you have 2 CO2s being made. And then lastly, you also made a molecule of ATP. So there was some energy here available to make to turn ADP into an ATP molecule. And that's what's going to happen here as well in this cycle. So ultimately, and I keep tracking your notes, here's what's made during the Krebs cycle. So in each Krebs cycle, right, you're going to take acetyl-CoA, join it with oxaloacetate, and form citrate. And then in the cycle, the citrate will ultimately become oxaloacetate again, right? Which is why it's a cycle, because then it can happen again and again and again. And in that process, the Krebs cycle, in every Krebs cycle, you make 3 NADHs, 1 FADH2, 1 ATP, and 2 molecules of CO2, which explains why we go from 6 to 4 carbons. Okay, that's a lot. You guys got to memorize. That's what's made during the Krebs cycle. Now over here, all those numbers I just said are doubled. The reason they're doubled is because this is going to happen twice, right? So you had not 1 molecule of acetyl-CoA, but 2 of them, because from each pyruvate, you're going to end up with acetyl-CoA. So we have 2 acetyl-CoAs. So this is going to happen twice. So you're going to do the Krebs cycle. Acetyl-CoA comes in, does the Krebs cycle. You make all this stuff, and then another acetyl-CoA is going to come in and do the cycle again. So the cycle, if you're starting with 1 glucose, which ultimately at the very beginning of these video, we started with 1 glucose. We then had 2 pyruvates. Those 2 pyruvates became 2 molecules of acetyl-CoA. And if you have 2 molecules of acetyl-CoA, you get to do the cycle twice, which means you're going to make not 3 NADHs, but 6 NADHs. Not 1 FADH2, but 2 FADH2s. Not 2 CO2s, but 4 CO2s. Not 1 ATP, but 2 ATP. And so that's ultimately what's happening during 2 cycles of the Krebs cycle, is you make all of this stuff right here. You make a lot. Okay, there's a lot of stuff going on in the citric acid cycle, the Krebs cycle. And then those ATP that we made were also made through substrate level phosphorylation, that process that I explained earlier, where there was an enzyme taking a phosphate and transferring it to ATP. Just know that that's how this ATP was being made. Because in the next video, we're going to make a lot more ATP, and that's going to be made a little differently than substrate level phosphorylation. But anyway, that's what's happening in the Krebs cycle. And then one last thing I'll note here is that if you look, there was actually some water that we used. We used 1, 2, 3 molecules of water in the Krebs cycle, and that's going to happen twice. So there's really 6 molecules of water that we are going to use during the Krebs cycle. Just remember that for now. You don't even need to remember it now. It'll be important in the next video. You'll see, because we're going to add everything up. But just know that there was 6 water. I just want to point it out now that to do this cycle twice, it required 6 water molecules. So in summary, for the Krebs cycle, again, I'll just say it one more time for you. You are taking acetyl-CoA, which is joining with oxaloacetate to become a 6-carbon molecule called citrate. And then citrate will go through this series of chemical reactions, turning back into oxaloacetate. And in that process, it makes 3 NADHs per cycle, 1 FADH2 per cycle, 1 ATP per cycle, and 2 CO2s per cycle. And then the cycle is going to happen twice. So then you can double everything. If we're starting with 1 glucose, the cycle would happen twice. And then that's all of that, the first 3 stages. So that's the glycolysis, pyruvate oxidation, and the Krebs cycle. I highly, highly, highly suggest you start like trying to put this in your head instead of just watching a video and pretending like you know it all. You don't. You need to like sit down, make a flashcard or something, and kind of start memorizing overall what's happening. And you'll see it's not as miserable as you think once you just memorize the basic gist of what happened in glycolysis. From beginning to end, what did we make? Pyruvate oxidation, from beginning to end, what did we make? Krebs cycle, from beginning to end, what happened just now? Like just memorize, and there's a lot of different components, but I promise you, many, many students, thousands of students before you have taken this class. Thousands of students will take it after you. They all have to do it. They all can do it. You can do it too, okay? Because at the end of the day, this is not even like, you're just learning like 5% of all of this. Like really, you'll learn in college that there's a lot more memorizing you're gonna have to do about all of the different reactions that I just breezed through. So then from here, I just want to kind of emphasize one last thing. We actually just got rid of all the carbons now. I don't know if you guys were paying attention, but we started this whole thing with 6 carbons in glucose. And then ultimately, we now have taken all of those carbons and we've completely broken it down into CO2 little by little. In pyruvate oxidation, we took away a carbon as CO2. We took away another carbon as CO2. That was 2 CO2s during pyruvate oxidation. And then in the Krebs cycle, ultimately what's happening is we're taking acetyl-CoA, which is going into the cycle, and then it's going out of the cycle basically as CO2 and CO2. We're putting this guy into the cycle, and we're just in the cycle in a complicated way. We're just separating those 2 carbons and releasing them as 2 molecules of CO2. So that's another 2 molecules of CO2. And then another acetyl-CoA goes in, and then that's going to be the last 2 CO2s. So really, at this point, we've made 6 CO2s, each containing 1 carbon. Those were the 6 carbons that we started with glucose. Those 6 carbons of glucose, we can say goodbye to them. They now are CO2 molecules. Okay, like all those carbons have successfully been broken apart into carbon dioxide. And really what's left now is just the electrons, all those high energy electrons that we stole from glucose. That's the only part of the story that we need to finish explaining in the next video. Like what happens to all these high energy electrons? We took high energy electrons in glycolysis and NADH, he's holding onto those high energy electrons. In pyruvate oxidation, we stole some. In NADH, there was 2 of them that did this. And then in the Krebs cycle, we stole a lot, NADH, FADH2, FADH2. And so where ultimately that's 12, if you add it all up, that's 12 pairs of high energy electrons that are now being held onto by NADHs and FADH2. Like what are they gonna do with these high energy electrons? Because that's really all that's left from the glucose. The carbons are gone now. Like, it's all CO2 now. And we just have those high energy electrons. And that's where we're gonna, that's the cliffhanger, okay, until we get to the final stage, the grand finale, where we'll see what happens to all those high energy electrons that we stole. But that's it. I hope that makes a lot of sense. I hope you're not too overwhelmed. If you just take the time and start putting it in your head, hopefully it won't be so overwhelming. In class, we're gonna talk about this all over again. We're gonna actually draw it all out together, some of the important pieces. So hopefully that will help you remember as well. But that's it. Thank you guys. See you later.